Okay. Yeah. So, well, to start with, one doesn't need to look very far to see how life has been impacted by advances that have been made in the life sciences and in biomedicine. It's all around us. So some examples here, if you consider childhood cancer, 50 years ago, you had a 5-year survival rate of 25% 5-year survival rate. So only 25% of children would survive. Now it's 84%. These are figures from the UK, similar figures around Europe, around the world. So survival rates have really dramatically changed through scientific advance. We all know how rapidly vaccines were produced, new methodology to produce vaccines at the beginning of COVID-19, very rapidly available. Now, of course, politically, it may not be that the distribution systems were as they should have been, but certainly the science was able to deliver some really important medical advances. We have a host of crops. The world's food supply is made more secure by technology advanced by life sciences that allow us to understand and to breed in more intelligent ways, to build crop lines that are resistant to drought, to adverse conditions. We have a biodiversity crisis. We have changing environments. We have climate change. Monitoring, understanding, and using science in policy making to know how to respond to such things as climate change is critical. The tools for this come from the life sciences. And then, of course, in the world of biotechnology, our food production here, the example brewing, has been transformed by the life sciences and our understanding of the organisms and enzymes that can be used. Some 80% of brewers use technology derived from the life sciences directly. And all of these things, they only happen because you have data sharing. So the data underneath the science are really key. And looking from on top at these advances, one can easily miss the fact that actually scientific data and the sharing of scientific data are really the lifeblood of these kinds of advances. So we're all stakeholders, we're all impacted by this. So what I'd like to do is talk a little bit about the importance of data and why what kind of data we consider and why, how the scientific world has responded to the need to share data and to make data available, and then how over the last decades really data has become a more important player in science and hence in society. And then I want to finish with some of the challenges that we face, because although the data are extremely important, they are not assured. We have some risks that we need to be aware of and we need to face. So I'm going to speak for something like 30 minutes, maybe just over 30 minutes. And then we have an opportunity for any comments or discussion points or questions there might be. I ask you, there should be a Q&A, question and answer button on your Zoom window. So if you put anything in there or raise your hand, let us know in there. I think my colleague Chuck Cook will be able to pick those up and make sure that we try to address them. So science advances in a cyclical way. So here we're looking at a typical cycle of science, but it's continuous and it's multiple. So let's start with a publication. So scientists publish their work in the scholarly literature. They publish a paper in a journal, and that's made available for other scientists to read. Based on that, other scientists build up their knowledge of a domain, and they ask their own questions. They build experiments and they run field observation, for example. Those experiments, those observations lead to data sets. They produce data sets. Those data sets are then interpreted by those scientists, and those scientists then author their own papers in the scientific literature, and they broadcast there and disseminate their research. And then so it continues. Science builds upon existing science, reuse of existing knowledge that's published in the papers, reuse of existing data. And so it's a very healthy system, but it requires this continuous communication at the level of the interpretations in the papers and in terms of the sharing of the data. So when we think about life sciences and biomedicine, we look at different scales of biology. We look at the very fine scale at DNA. We look at genes and genomes. We look also at proteins. We look at biochemistry, the way biochemical pathways take place. We look at larger scale, we look at cells in organisms, we look at tissues, so we look at blood and we look at livers and we look at leaves on trees, and then we look at whole organisms. And beyond that, we look at ecosystems, so multiple individuals, multiple species interacting in an environment. And so biology covers all of these, and all of these areas are important. And we have data that relate from different types of experimentation and observation, data that relate to all of these different types of biological entity. I'm going to give a couple of examples, and I've chosen these. I'll come back to these two examples as I go through the presentation. So this is on the left here, we have a picture of DNA. So if you had a very powerful microscope, this is what DNA would look like. It's a long string of a molecule, and it encodes information. So the function of DNA is, there are many functions, but essentially it encodes an instruction set for an organism to build itself and for it to operate itself. So it's really the code of life. And so this is a DNA molecule. That's what it looks like. But actually, the information content it's all embedded in chemicals that lie within these ladder-like structures between two strands. But essentially, if you read the sequence of a DNA molecule, it looks like this. It's a string of letters. It's only 4 letters. So it's a very simple code, but this is only a small section of sequence. Actually, it's much, much longer. So that's what data look like for sequences. Here's another example. This is a protein structure. So proteins are the functional units of life. So living systems do things because they have protein. So if your muscles contract, that's happening at the level of proteins. If a signal is coming from a nerve signal is transmitted across into your brain, proteins are involved in that. If you have a skeleton, proteins are involved in producing that skeleton and sustaining that skeleton and make up the skeleton as well. So these are the structural and the functional components of living organisms. And so this is what a protein looks like if you were to zoom in very closely. It starts off as a string. It's a very long string of a molecule. But as it matures, it folds up, and it folds up in a very, very back in on itself in a very, very specific way. So you could stretch this out; it would be a long string, but actually, it's a very it serves its function as a very folded structure. Now, the folding is really important because it's that's what determines the shape, and it's the shape and the faces that it presents to the rest of the world that make a difference to its function and what it can do. So scientists care a lot about the shape of proteins. They care about these structures. So that's what it looks like on the left. But the data set that's derived from that, you represent that by a set of coordinates. So here we have a table of coordinates that define where the parts of the string are and how they've folded up in space. So that's a protein. So there are 2 examples of data types. Now, a characteristic of most life science and biomedical data is that individually, a data point is not particularly informative, and often it's not very informative at all. Here's an analogy. You imagine a telephone system. So I've been, and many of you will have been, to visit a country house, a country estate somewhere, and you learn on the tour that this was the first household in the country to have a telephone. And that's fantastic. But of course, the obvious question is, well, if you're the first to have a telephone, what do you do with your telephone? It's a great concept, but if there's no other telephone, who do you speak to? So although it's a great concept, it makes sense only when there's more than one. So when you have 2 telephones and you connect them, it's a valuable system. If you have many more telephones and you connect them all together, you have a useful network and you can communicate. And then, of course, we all know that the world is hugely connected, and one can communicate all around the world, and it's a very impactful system. So the actual infrastructure, the value, comes when you have these multiple entities that are all connected, and it's very similar with life science sequence data. So on the left, this is our sequence again. This is a gene sequence. if you were to show this slide to a group of experts, a group of biologists who look at these things every day, they're very unlikely to be able to tell you anything about that sequence. It's really not valuable or informative on its own. But if you have something to compare it to, then it becomes really useful. And if you have a whole collection or a whole database of these sequences from different experiments, from different organisms under different conditions, different scenarios, then you have a really informative data set that can be really useful and can lead to all sorts of impacts. So it was with great foresight then that a previous generation of scientists to me back in the early 1980s understood that actually sequencing, which was then an emerging technology and quite a niche activity, that it was going to become very important. And there was a need for scientists to have a way in which they could share their data such that everyone could pull out the maximum value, building sets of connected sequences into a big database. And so this was started up the hill over Heidelberg at the European Molecular Biology Laboratory. And they built what was called the EMBL data library. And this was a database that gathered the world's sequences from scientists who would send them in or otherwise provide them. And they built the first database. And what we're looking at here on the left at the top is one of the design documents back in the early 1980s, designing the data structure that was ultimately used to build the database. So very quickly, a group in the US began activities in a similar way, and so EMBL collaborated with them, and soon afterwards, a Japanese group was doing the same thing. So a three-way global collaboration was founded called the INSDC, and this is a collaboration that's much celebrated, where on a regular basis, each of the databases presents its new data and shares it with the other partners. So you have you share the work of capturing and organizing the data, and you provide a broader coverage. So many things happened. There are all sorts of useful things and many technical advances. Sequencing has changed since the 1980s. A good demonstration of how this was useful during COVID, but many other examples. And actually now the collaboration is open to further organizations to join from further parts of the world, and we're working with groups that may become new members. But essentially the principle is the same, that you have a value in organizing the data and collecting them together and making them available to scientists around the world. And that principle, although it looks very different, this is what it looks like now on the bottom right here, the principles are the same: collecting data, making the data more useful in that way. So actually, that was just sequences. Other data types from other parts of biology were at the same time becoming more important. New databases were forming, and it became very clear to EMBL that actually, this was a whole institute's worth of activity. So a new institute was founded, and this is the EMBL European Bioinformatics Institute, that was founded as an outstation of the main lab in Heidelberg. So it was founded in 1994. It's highly international, collaborative, innovative, and interdisciplinary, and really a global champion of open life science and biomedical data. It does lots of things. It has an active research program around computational biology, around the data. It trains people around the world to use tools and services and use the technologies. It partners in various ways with commercial players in industry. But what's particularly relevant here is that it provides very substantial global data resources, so databases and services that are used continually by people around the world. It provides some of the world's largest life science and biomedical databases, and it provides them very typically in collaboration with different groups doing similar things in different parts of the world. And so the rate of usage is really quite substantial. So on a daily basis, we have 107 million hits, 41 million unique users. That's in the time frame of a year. So these are really big numbers. So second to the Googles and the eBays, these are massive numbers for people hitting accessing services on websites. We've seen really remarkable growth, and so these are plots. These are all on different scales, but back here, we're looking at 2004. We're moving towards the present day. What you see is not just a growth. We're looking at the numbers here, not just growth over time, but you see growth of increasing rate. So this is doubling every, sometimes with sequence doubling once every 18 months. So that means that if you look at the database after 40 years, most of what you see in there, the majority of what you see in there has just come in the last 2 years. So this is really rapid growth on growth. And you can look at all the different data types, and it's the same story, that technology is advancing, more people are using it, it's becoming more useful to more people, and things just grow and grow. And so I can show you a shot of, I think I can show you, if I still have it, a shot of live hits on the EBI website. So what we're looking at here is different users, so they're organized by where they are in the world, accessing things from the EBI website. It could be software accessing, it could be individuals accessing. So you see a big cluster at the moment in the middle of the working day in Europe. The numbers in the US will gradually grow. What you see actually is, as the Earth moves through the sunlight through the day, you see a sort of shift from east to west as more people get working. But so these are real numbers. This is here and now. It's just a constant access. So these data are really important and really used around the world. So because of the numbers, because of the scale, there's a great opportunity that's being leveraged to use modern data science methods. So we hear a lot about artificial intelligence and machine learning, and these are becoming particularly important. So life science and biomedical data are well structured and well organized, and at a large scale. So very appropriate for training artificial intelligence models, for organizing the instruction set that you use to allow machines to learn. So researchers and data scientists who develop algorithms are using data at scale from life science sources such as EBIs. There's a lot of curation around the world, a particular focus on that at the European Bioinformatics Institute. So people are expertly curating, organizing, and structuring the data, making things consistent, adding value by annotating. And then AI learns things, and where appropriate, we can feed back the knowledge from AI back into the system to enrich the records and make new databases even. And so a great example of this is the AlphaFold system. So this was a collaboration between Google DeepMind, EMBL EBI, to build to use AI to solve one of the major challenges that biology has faced over decades. And so it's not possible to use this without training. So we had data at scale, well-organized data from structural biology. So I showed you a protein structure. We have the sequences that I showed you as well. And it's the most recent release has now over, so that the challenge is that you need to understand, well, until AlphaFold, it wasn't possible to predict how that string of protein folds up into the very specific, very meaningful shape. You had to experimentally determine it. But what AlphaFold does is it gets around that problem. It allows the machine to calculate the structure based on the sequence. So it tells you how a particular protein will fold. And so we now have over 200 million protein structures available for scientists to use, openly available for scientists to use. Prior to AlphaFold, we had 1,000 times fewer. So this is a massive, massive step change, and it really serves to accelerate the science people are doing, and it brings us closer to the kinds of societal impact that I illustrated before. There are many potential applications of AlphaFold. One is in, of course, drug discovery, design of new kinds of drug, vaccine development, useful in all sorts of areas, infectious disease, neglected disease. In biotechnology, dealing with perhaps ocean plastic pollution, dealing with food technology challenges, new tools, new industrial processes, and so on. So great opportunity really to accelerate what science is doing. It places a big, I'll come back to this, it places a big demand on the data and the people that support the data, but it's extraordinarily powerful. So I mentioned these as examples of benefits and that come to society through data and through the science that uses those data. And I'll just go into a little bit more detail on a couple of examples. And the first one is a COVID story. So on the left, we're looking at a molecule. This is a compound called molnupiravir. It's a drug that was repurposed very rapidly during COVID. So there wasn't an obvious frontline therapy to treat incoming COVID patients. And this was one of the drugs. So repurposed, it already existed, it was already used for other viral disease, but it was repurposed specifically for COVID because it was to be effective. And so the repurposing happened within months, and it could happen because there were data sets that were openly available to the scientists looking to develop molnupiravir for this. So we had structural biology, we had sequence data, we had other databases that were available. And without those databases, it possibly wouldn't have been repurposed, but it wouldn't have been repurposed nearly quickly enough to have an effect. And the completion of the story is that actually further data since its use show that actually it's not the safest of options. There are now better alternatives, and so actually it's largely been replaced. It's not used or not used as frequently as a frontline therapy. But that's a whole cycle of use and maturation of a therapeutic approach that is only possible on a usable time frame because of underlying data and the fact that those data are available to scientists. And here's a second example from the environmental biodiversity world. And it's the example of the plankton and understanding their dynamics. So plankton are those organisms that live in the upper layers of the oceans, and they float around. They're typically quite small. They float around with the currents and the various flows. We think about the rainforest as being the lungs of the planet, but actually more than half of the world's oxygen, the greatest part of the world's oxygen, is produced from this layer of the oceans. So the things living in the oceans are hugely important for sustaining a healthy atmosphere for the planet. In terms of carbon capture, they capture something like 40% of the global total. So they capture carbon dioxide from the atmosphere. It's used in their bodies to make them, and when they die, they sink, and there's a huge sink at the bottom of the oceans of sequestered carbon that's very long term. So it stays locked away in that form for a long time before eventually it recycles. So a really important service to planetary health. So we care about the plankton. We have to care about the plankton. And of course, they're sensitive to climate change, and they're sensitive to other environmental change. They're sensitive to pollution. But to understand all of this, we really need to know what the diversity is. We need to know which species are there, how they live, what their sensitivities are. And so to do that, there's a great deal of marine science going on. And this is an example with which EMBL has been very involved. This is a ship called the Tara. The project is called Tara Oceans, and Tara sails around different voyages, sampling and understanding the diversity, collecting data, understanding the distributions of different species and the changes in those distributions. There's a more recent recently launched project called TREK, which is traversing European coastlines. So a deeper study of pollution in coastal waters, understanding the diversity, understanding the impacts of human activity around the waters as well. And so really, none of this works without this kind of data collection. And these are the data that really give us our baseline, and they give us the opportunity to monitor how things are changing and to change policy, to change the way we handle issues such as pollution, the way we handle issues such as climate change. So all of this is really great. Science is really, life science, biomedical science are really impactful on human life. There's a lot to come. Science absolutely depends on data, but the data themselves, or the long-term security of the data, alas, there are risks. things are very fragile. And that fragility really arises from the way in which people have been running the databases and collecting and sharing the data. And so really, global life science data are make up an infrastructure, or the databases that organize those data make up a global infrastructure. But we don't think of it as an infrastructure, and it looks very different to other scientific infrastructures. So perhaps when we think of scientific infrastructures, we think of a telescope, or an astronomical telescope, or we think of a high energy physics facility. So here we have CERN, so high energy physics. So here we're looking at a particle accelerator. We have a physical infrastructure. So this is a ring. This is the LHC. It's a ring that runs under Switzerland and France. It has buildings and sensors and instruments in a number of locations that emerge from above the Earth. But this whole system was designed from the top down. So governments got together and they planned out a big experiment. They'd build a collider, and they would operate that collider and run certain experiments. And so from the top down, they designed things. They brought the scientists together, and everything was planned, and everyone knows the parts. And there's a building phase, an implementation phase, and then there's an operational phase. By contrast, on the right-hand side, we have, as far as we know, more than 3,000 different databases that are serving useful life science and biomedical data to scientists around the world. And we know that these are important. We know a lot about a small number of them, a little bit less about most of them. But there's never been any top-down organization. It's always been individuals in institutes understanding the importance of a particular data type and starting up databases in a very ad hoc way. So the system is very organic. And the result of that is that there's no one knows exactly what's in the system. Everyone knows it's useful and they know their local parts. But actually there's no top level description, and there's no long-term planning for how you sustain this and how you get the most value from it. And so there are some significant sustainability challenges. Some of the databases that are really important to scientists around the world have no long-term, many of them have no long-term plan and no long-term sustainability. The funding that comes in, it typically comes from the research funders, the life science research funders. It's been haphazard, often very short-term, just a few years. These things need to run for decades, and very unevenly distributed around the world. And this is because there is little global coordination around the provision and the sustainability of these resources. So because of the demands of AI, because of the advances in technology, the increased effectiveness and output of the existing technologies and new technologies coming online, the system is an already challenged system, is increasingly challenged. We have great opportunity, but unless we really work to fix the sustainability issues, then we will see components of the system dropping out, and we will be able to realize less of the value, ultimately impacting some of the things that society would have benefited from. So this is where the Global Biodata Coalition comes in. So the GBC is a coalition of life science and biomedical research funding organizations. So it includes government research, government ministries, research agencies and councils. We have a couple of foundations in there, but actually we seek to have many more organizations, including of different types in there as well. But it's a coalition of funding organizations, and they come together recognizing the importance or the essential nature of the databases, the data resources, and the data they contain in pursuing their research programs. So they know that the research programs they fund simply won't deliver if the data aren't sustained, if scientists don't have access to the data. They understand the perilous situation that we find ourselves in. They understand the risks, and they acknowledge that there is a real problem. Things are very fragile, and then they commit to. They understand the need to act, and they commit to trying to find ways to bring about a better situation. So the GBC is there really to bring these groups together, to enable them better to coordinate, so better international coordination, because it hasn't really existed in the past, and then to ultimately ensure a better way of sustaining support for the for the infrastructure such that science isn't disrupted and society isn't disrupted. They do this work in many different ways. Essentially, it's about bringing together stakeholders. So we bring together the people that run the databases, we bring together the funding organizations, we bring together the scientists that use the data to really understand what a sustainable system looks like. We spend quite some time profiling the landscape. So I said we know a lot about a small number of databases, but there are many databases that we have them on the radar, but we don't really know so much about them. And we're not in contact yet with the communities or the people that run them. A lot of time is spent landscaping and then working with the stakeholders, understanding what we can agree and what we can work towards and what the mechanisms might be. And so, for example, we're just completing work on a big consultation that we ran earlier in the year, or finished earlier this year, where we look at some of the principles that we need to follow to build a more sustainable system, and we look at what opportunities there are, what kind of models we might start to investigate to do that. So everyone in some way is a stakeholder in data, in life science and biomedical data. And so you may be an individual, you may be an organization, you may be in a company, you may be a government. In some way, life will be impacted by something that comes from some advance that has been supported by life science or biomedical data, be it health or the environment or whatever it is. And so for those of us one of our priorities really is to raise awareness of this issue. It's great that if it's not particularly known, it would be great that people know about the issue, that they communicate, they talk about the issue, they bring it into the conversation. And many people, particularly in this audience, will have influence in different ways, perhaps over policymakers, perhaps over business decisions, perhaps over political decisions, all sorts of different things. There will be opportunities to influence and raise awareness about this really critical infrastructure that is at risk. And so I invite you to have a look a little bit more at the highlights and the impacts from one of the providers of these resources, EMBL EBI, and then to look a bit more, if you wish, about how we work with the Global Biodata Coalition.